Welcome to our BroadE workshop today. I'm delighted to see you all. So the title of our workshop today is From Missense Variants to Protein Sequence and Structure. This is the quick overview of today's workshop. I will first go through the problem and background. This will be a very short lecture, but hopefully useful. And then for 60 minutes, we'll go through the first tool, PER, a pathogenic variant-Enriched region, and then we'll have a break. The second half will be on MISCAST, Missense variant to protein structure analysis suite. And we will have another break here too. So starting with the problem and background, first the problem. So the problem that today we'll be working on together is genetic variant interpretation is demanding. As we all know, probably being part of BroadE, that human genetic variation data is exponentially increasing every day, and the challenges in their interpretation are growing also in parallel. And this challenge is both in clinics for the test interpretation and for patient counseling, and also these challenges exist in laboratories where variants needs to be selected for molecular experiments. So having said this problem, now I will go a little bit back and will show you that what are the genetic variant types. Now this is a normal condition where from DNA level to mRNA level to protein level, you can see that we both have 2 copies of gene and both are good. So we have both copies fine, and we have both protein structure for both the copies. So everything good. Now, what can go wrong? So first type of wrong thing can happen is by changing a single nucleotide in the genome that we call as single nucleotide variation, or SNV. And this is a variation type that may or may not change an amino acid in the protein sequence. Now, when it leads to the change of amino acid, then we call this variant as missense variant. And missense variant changes the amino acid in a single position, so a single position on the structure is changed. Now, the second type of wrong thing can happen in this way, that we have a sudden stop codon happen in our genome sequence, and because of that, 1 copy of the protein is gone. And so we just have 1 protein structure available and the other copy is not available. Another type can be that the whole gene is deleted from our sequence. So there is no copy of the mRNA, and of course there is no copy of the protein. And next, what can happen is we have too many copies. So too many mRNAs of the same gene and too many structure for the same gene, right? Now, given that now we know that what could be the variation types or variant types and what could be the consequences of those variants on proteins, let's ask ourselves a question: what fraction of the human genome represents proteins or protein coding sequence? We all know the answer, right? Yeah. So it's very, very less, right? Only 1.5% of the whole human genome sequence is codes for protein. And the rest are non-coding RNA molecules, regulatory elements, and sometimes some repeated elements and sequence for which sometimes we do not know if they have any particular function, but they may have. But what we know is this 1.5% of this sequence that codes for protein performs most of the cellular function. Now, knowing this, we can guess that for these 3 types of genetic variation, we know that either there are less protein or more protein, right? So we can have an intuitive guess that something will go wrong, probably. However, what we cannot confidently tell is what will be the consequence of this type of variation, because this changes 1 single amino acid, and it can be benign, it can be pathogenic, it can be somewhere in the middle, and we will learn more about it in the coming part. So this adds the second line in our problem statement that 50% of the reported single nucleotide variants in ClinVar, which is one of the largest clinical variation storing database, reports 75% of all SNVs as missense variants for which there is no significance known, which means we don't know that if they are benign or if they are pathogenic, right? Now, from that, the third dimension in our problem is missense variants are particularly hard to interpret. That is, it's particularly hard to tell if it will be benign or pathogenic because almost every disease-associated gene has both types of variants. Some amino acid changes are benign, some amino acid changes are pathogenic. So we never know. Now, given these three problems in hand for variant interpretation, in our group, we made some efforts to interpret the amino acid changing single nucleotide variations or SNVs on protein sequence and on protein structure. So here you can see that a single nucleotide is changed possibly here, which propagates in the protein sequence here, and that propagates to here in the structure. So we would like to interpret the impact of this variant in the protein sequence and in the protein structure. So having said that, now I will welcome our invited speaker, Eduardo, who is a staff scientist in Cleveland Clinic. So welcome, Eduardo.
Good afternoon, everybody. My name is Eduardo Perez. I work in Cleveland Clinic in the Genomic Medicine Institute. And today I'm going to talk to you about the identification of pathogenic and variant-enriched regions, which is a novel tool for variant interpretation. I come first before Sumaiya because all of this work is in a two-dimensional way. It's on the linear protein sequence, and then Sumaiya will move everything into a 3D modeling cool stuff. So I will put this into the context of variant interpretation and in the how can we use this in agreement with ACMG guidelines and clinical testing. So this can be directly applied to the ACMG guidelines as I will show you. So the contents of my talk, it's these bullet points, 5 bullet points. I will try to be brief on the things that I'm sure you already know. As Sumaiya was saying, missense variants are the most difficult type of variants to interpret because they can have a gain of function, loss of function, or even a neutral effect. But the important thing also to consider is that between two individuals, we can expect between 10,000 or 12,000 missense variants, in contrast to PTV, which we can have between two individuals 150 or 200. So on top of that they are the most difficult to interpret, they are the most likely to be found in a clinical test. So how to interpret variants and their relationship to disease? We use a lot this homology, which is pretty similar to how to solve a crime, because we have an accused, Mr. Variant here, that is accused of causing the disease of the person next to it. And then we have a judge, which will be us, that will decide, according to several arguments, if the variant is in fact causing the disease or not. Is the variant guilty or not? Sorry, this is the other way around. So the translation into the genetic part of this judgment is as follows, as the figures clearly show. We have some alibis that are arguments for benign variants or neutral variants, and we have arguments for pathogenicity. And these arguments can be are pretty heterogeneous. And at the beginning, when clinical testing approach to the patients, people address this issue from different ways. So at the end in 2015, and it's not the end, I'm sure they will come up with a new set of rules in the upcoming years, the scientific community and the clinical genetics community agreed on 28 criteria to for variant interpretation. And in combination, these arguments will allow us to make a call, make a judgment of the pathogenicity of the variant that we are currently studying. These are the 28 criteria that ACMG guidelines possess. I do not intend for you to read all of them right now, but it is important to acquire that we have very strong arguments for pathogenicity, then strong arguments for pathogenicity, all the way down to only supporting evidence for benign variants. Sorry. These arguments, the ACMG guidelines, in several combinations can call pathogenicity or neutrality. So it's not enough to have just a very strong argument to call pathogenicity. You need to have a very strong argument in conjunction to two moderate arguments, to one very strong argument, and so on. These combinations, I will not show them because it's really difficult to see, and I will show you later on how to address this issue also. The important thing here is that there are several computational approaches that can be immediately integrated on top of these ACMG guidelines. Most of them are moderate. We have also supporting arguments and strong arguments. As we will see during this presentation, most of the tools and computational tools that we know are just a supporting argument. Here, the last one, PP3, multiple lines of computational evidence. Here is where most of the tools that we know fall: SIF, Polyphen, CAT score, GERP score, and so on. So even if we have like a damaging effect from a computational tool, this is not very useful when it comes to variant interpretation on its own. So we cannot use these tools on their own. Computational approximations can be summarized in two main categories. The ones that address the biological context, that means conservation, structural features, domains involved, and here are most of the scores that we know. These scores are usually aggregated on tools like ANOVAR or BEP, that I'm sure that most of you will know. And then we have the frequency context, which by themselves are far more strong arguments for variant interpretation than the other computational tools. We have the argument of the general population. That means if we have a variant in the general population, it's less likely to be pathogenic. Then if my variant is present in another patient, it's most likely to be pathogenic too. So these are the frequency arguments for, that can also be addressed computationally. I will briefly describe ClinVar and NOMAD. As I already mentioned, ClinVar is a patient variant repository that addresses the relationship between variant and disease. And as I said, if a variant, the exact same variant, has been observed in other patients, I am pretty sure that my patient will have the same state too. Currently, it's the largest patient variant repository. It's not the only one. There are several ones with different approaches and advantages. But I can say that this is the largest and the most popular one. Currently, we have like 500,000 variants on ClinVar.
Oh, I just had a question.
Sure.
Is it specific for certain diseases in ClinVar?
No. People can submit, and anybody, that's really important, anybody can submit variants to ClinVar. So, and the most bigger submitters here are clinical testing companies. So they are the ones that submit these variants. So no, and a lot of them, the phenotype is not provided. We just know that it's pathogenic or not. So many cases, you just know that that variant is found in a person with a disease. So it can be really of low confidence, the information there. It requires a bit of cleaning. Then Oh, I'm going backwards again, sorry. Then NOMAD, which is a repository of the general population. We have nearly 150,000 exomes and genomes. And the argument here, as I said, is if the variant has a frequency on the general population, it's less likely to be pathogenic because these people wouldn't be recruited as controls in all of these studies. So as a summary, coming back to the ACMG guidelines, we have ClinVar and NOMAD as a strong and moderate arguments. And then we have all of those computational tools that, that measure the damaging effect of the variant in the protein just as a supporting argument. So I want to focus briefly on one measurement that we developed at the laboratory, which is the paralog conservation, which is a measurement of the conservation that has a specific amino acid. And it's pretty successful when it comes to studying neurodevelopmental disorders. A large number of neurodevelopmental disorders are related between each other. This is self-evident when you evaluate the epilepsy-associated genes. We can see that most of them have similar names. They belong to the same family. And when we say that they belong to the same family, we're saying that they come from an ancestral gene. But this ancestral gene is human, and all the family members are genes that you contain on your genome, in contrast to orthologs that are genes related to each other but from different species. So here we can leverage this information and measure how conservated is an amino acid, and this is pretty useful for variant interpretation. So in the context of these gene family members, we can align their sequence, and we will most likely find regions that are conserved in the, do you see the red dot? Kind of, no, not much. But you will see regions that are conserved here, denoted by the dark colors. So they are all the same between paralogs. And then you will see regions that are not that conserved. You can assume that, this one? No.
Because you are old.
Okay. Thank you. No. But this goes backwards. Okay, yeah. Okay. Yeah. Coming back to the alignment of the paralogs, the genes here that are related to each other, we can expect that the regions that are conserved between paralogs are really important for the protein function, and thus can be related to disease because they're sensitive to variation. And then the regions that are not conserved, they're less likely to be important. So another important fact is that 80% of disease-associated genes actually belong to a family, to a gene family. So we can examine a lot of gene associations with this paralog approach. So we develop a score for that, so we can tell if with the number how conserved is the amino acid. But we wanted to push this a bit further, and in combination, we decided to address the identification of pathogenic enriched regions, which is the tool that I'm going to show you today. Can we combine different references databases that I just mentioned to gain biological knowledge? We made 3 questions here. Are regions within genes intolerant to variation? We know that there are a set of genes intolerant to variation. They're really sensitive to variation. We can explore this on NOMAD already. But can we subset that to smaller regions of the protein? And then on the other side are disease-sensitive regions within the same genes. Are these anticorrelated? And if this region exists, wouldn't they be shared between paralogs? So we decided to take these 3 tools for variant interpretation and combine them to see if we can gain knowledge. And we will try to identify these pathogenic enriched regions. Coming back to the paralog alignment, where would you expect to see an enrichment of general population variants? In the non-conserved region, I will say, right? And then where would you expect to see patient variants? In the conserved region. So what we did is to actually try to prove this concept and map all pathogenic variants coming from patient repositories of confidence, of course. No variants of unknown significance are included here. And we map them on the alignments. And on the other side, we mapped all the general population variants here on the same alignments. We did it in a way, since these two sources of variants are way different, and we have coverage here, while here sometimes we don't have method for sequencing. So we decided to take a binary approach, yes or no, is the amino acid mutated or not? And the alignment of these variants look like this, a matrix of 1 and zeros with an expected behavior that we wish it was like that. So we will see constrained regions here that are conserved and with absence of variants from the general population. And then an enrichment of patient variants, pathogenic variants within these regions. This is the ideal case. And once we have this mapping, we can perform a burden analysis to evaluate the load and come with some sort of burden graphics across the linear sequence of the alignment that will be valid for all of the genes involved. So this burden analysis, when we find this anti-correlation between constrainment from NOMAD and enrichment from patient variants, we will call a pathogenic enriched region. This is the ideal case. So then, I'm confused again with the, okay, so we have this, this is what I wanted to say. We have the gene family set of analysis, but during this process, we also realized that we could do the same for genes alone. So just compare the burden that you can see on one gene from NOMAD and the gene from, and the same information on the gene for patient variants, and we can do a gene-wise approach. This, of course, is less powerful, but also will enable us to evaluate all the genes in the genome with this approach, and not restricting the results to only genes that belong to a family. Overall, we map all genes. We included more than 2 million missense variants from NOMAD and nearly 100,000 pathogenic variants from ClinVar and HDMD, 10,000 alignments almost, and 2,800 gene families. All of this was gathered together in a framework of Shiny, that is our way of developing apps to deploy on the internet. And these results are available at this address right now. So overall, we found more than 40,000 amino acids involved in these regions, and also with 1,200 genes having at least one PER. How yes?
How do you treat paralogs that have like insertions where you can't align them across family?
Right. When you have like a gap for a certain gene family member, we punish that because we assume that it's not conserved on the other family members, so it's just present there. Of course, this comes with a small sacrifice because some regions that are paralog specific, let's say, gene specific, could have certain involvement in diseases, but we were searching for something more common between the families. So when we map those regions as ones, when it wasn't aligned, like when you have a gap on the alignment, we assign it a 1 on the matrix that I showed you before. So as it is if it has a normal variant. This is how real results.
Is the method robust to making calls not binary? So for example, if you use a score of amino acid substitution conservation that penalize big changes more than little ones to make it continuous between 0 and 1? Would the downstream analyses break?
We tried, when we were developing the method, we tried several ways of, because the method that I'm showing you now includes all missense variants. Right. So variant.
Any amino acid change, but
yeah.
some are worse than others.
Exactly. So this will have a larger effect. But when we increase the bar, we lose some results, and we decided just to include all type of amino acids. And this is because we're not measuring the amino acid exchanges itself. We are accounting for a region, a position that is sensitive to disease. So whatever happens there will be prone to be pathogenic because of the position rather than the exchange.
Yeah, I think you might even clean up our signal if you give less credit to,
yeah.
leucine, isoleucine, than something else.
Yes, it's perfectly plausible. This is the voltage-gated sodium channel family. We have all the SCN channels from 1 to 10 here. This is a 2,000 long alignment of amino acids, and we found 16 regions pathogenically enriched. We can see that we have pathogenic variants distributed all across the sequence, but only these regions were found to be significant. This does not mean that these regions are not able to cause disease, but we're pretty sure that inside these regions it will be pathogenic. When you look at the results on a table, you can see one of the main advantages of this method is that it's able to accumulate variants on the same site. So we have disease-associated variants from different genes here on the same analogous site that it's conserved. And the other thing is that it's able to capture sites conserved that are surrounded by pathogenic variants, but when you look at it on a singular way, you will not see any reported disease variant yet. And we tried this with previous version of pathogenic variants, and we see that with newer versions of, with new CleanBar releases, we were capturing more and more sites that were expected to be pathogenic. So this can be directly used for variant interpretation. How the site looks like this. So you will have an input that can be any gene, and if the gene belongs to a family, you will see family-wise results like this. But you will have, and here you will see the family plot, which is the same that I showed you before. You have the nomad burden in green, the patient burden in purple, and the pathogenic regions in red. If you want, you can change to the gene-wise results. So all the time you can see the burden that it's observed on a single gene, but you can see clearly here that the burden is, the anticorrelation is not that clear. So showing how the family approach was more powerful. Then the user can zoom to specific regions, and all the time you can see below the table mode when you can be specific, and this is the part that is most useful for variant interpretation. You can search for amino acids and explore how the burden of patient variants is shown. Next to the table mode, you have specific features of each pathogenic enriched region. Not all of them have the same significance. There are some regions that are stronger than others. You can see which genes they are contributing to the signal, and the diseases that are associated to that specific region. So overall, how we can use PERs in the context of ACMG guidelines. We can consider it a mutational hotspot because it accumulates mutations on a conserved region, and it's already on top of a supporting argument. But of course, by itself, it cannot make the call of pathogenicity as any of these tools. So this is how we can use PERs. The paper was accepted last month. So it's already available if you want to look at the method more in deep. And now I would like to show you how it works. So, okay. So I invited you, and I hope it works, because the site is not prepared to have 50 entries from one IP. So, yes, but can you access can you see the browser? It works? Oh, pretty great. Yeah. Okay, cool. So, if you're all inside the site, for those of you who are more into bioinformatics, we have a bed file for all the PR regions. If you have 1,000 variants to make a call of pathogenicity, you can just use intersect beg tools to cross these regions and annotate PRs. But what I'm going to show you now is how to use the site for single variants and explore how we can extract information from this.
Yeah, sorry, I missed what you said about the batch mode.
Ah,
How does it work?
It's not implemented, but it's available. I have all the PERs on a genome-wide level, these 40,000 amino acids, on a bed format. That means chromosome start position, end position, and a long list of that.
You can't get that
From the website, no, you can get it from GitHub, where I uploaded all the PERs. So that's for massive annotation, and can also be implemented as a plugin on BP, for example, or cross it with bed tools.
GitHub link on the website?
No, that I can recall, but it's on the paper, I'm sure, or I can send it to you. But, so I prepared 3 examples. You will get from a genetic test the HGBS format of variants, that it's really ugly, and it's not very comfortable to even integrate into the analysis. But we can see here that this variant belongs to the gene green2A, and it's a valin on the position 632 that changes into a phenylalanine. So, since I already mentioned, PRs do not care about the exchange, just the position of the amino acid. So in this case, we only need to search for green2A and valin position 639. Any exchange there will be belonging to a PR or not.
Why is it 32?
No, this should be the same number, sorry.
And the bottom one, same thing? 15, 17?
Yes, you're totally right. This is the one that matters. Is anyone not in the site? Oh, that's great. I can show you a live also how to go there. You don't see the. Oh, it's not extended here. So, just wait me a second. Okay, do you see the site here? In the example that I was just showing you, it will be green2A. We first search for the gene. We select the gene and submit. This gene belongs to a family of 4 members. And the variant that I was showing you, it's green2A valin 639. Was that correct? So on the table mode, right below the burden plot, you will find the column from green2A. And yes, it was valin 639. So valin 639. Okay, this amino acid belongs to a PR4 on the burden plot. That it's right here. The interesting thing here is that the pathogenic enriched region here is showing you a disease association for another gene. that is analogous to green 2A. So if you look in ClinVar for this variant in any position of valin, let's say, you won't find any variant associated to this region. But with this approach, you can conclude that this is a pretty sensitive region to disease. So that's the first example. If we come back to the presentation, the second example is this one, is the gene SOX9, which this variant will be translated into SOX9 P128.
So I'm not getting the behavior of the bottom table that you just showed.
Yeah, I think it's just what
Yeah, it took a
it
little
just
while.
it takes some time.
It's loading.
Says no matching records found. Seems valid.
On green 2A, the underscore, yeah.
It's not coming in. It doesn't matter what I type there. I can try to
Oh, it's not loading, I'm sorry.
Did we crash it? Good job, guys.
It's highly likely.
Yeah, no, I
Came up? That's great. Okay, I'm relieved. So then SOX9 P128 underscore, that's important. So you come here, new search, SOX9, and submit. You see the burden again, and you search for SOX9. This is a pretty fast example, so watch P138. And this time, the variant doesn't belong to a PER, and I put this example in the presentation because I want to stress out that the fact that a variant is not present in a PER does not mean that it's not pathogenic. On the other hand, we are just really sure that the ones that are inside are more likely to be pathogenic. So if you're trying to deal with a variant that is showing this behavior, it's not a big deal if it's not inside a PER, but just that you can be really sure if you found someone inside, it's really good for you.
So on the x-axis here, which of the gene family members is it following in terms of amino acid sequence?
All of them. So this proline is present in all of the.
But I mean in the graph. So for example, with the PERs. So for example, is it? It says 0 to like 500 the x-axis. Right? Which is that most representative?
The positions here shown are from the index, which is the final count of the alignment. As you can see, these numbers, the alignment here is 138, but for each protein, it has a different index, a different position. So this is the amino acid position of SOX7 that falls in the position 138 from the alignment. The alignment is larger because it has gaps to make the proteins fit, so that's why it's always longer, and we call it index. So the specific positions that are real positions are the ones for each of the gene columns.
So it looks like in this case, for example, this variant falls in between PR1 and PR2?
Yes. Yes.
138,
Yes. So it does not fall into the enrichment, but you can see here there is a small rise in the general population burden, so that's why it's tolerated more. One important thing is that we calibrate this burden count is done by a window that goes through the alignment and counts and asks if the window is enriched for pathogenic variants or not, and when it finds significance, calls, and in this case, that window was calibrated. But let's say if I try a bigger window, that site will most likely be captured by the method. But which, yes?
Oh, sorry to interrupt. I was just going to ask, you may be getting to it, but for variants that are not in the PER windows, are there continuous scores that we can use from the pathogenic side or from the...
Yes, you can, for example, here on the same side of the paralog of the burden analysis, we also implemented the paralog conservation, which is just a measurement of how conserved the site is. If you click on that, you will see, well, all of these sites, and the one that I put as an example is also really conserved. We saw that all of them were proteins. And this score is available for all amino acid positions. So you can find a measurement for all of them. Here you can use CAT, GERB, CIF, all the other scores that you will get from tools like ANOVAR or BEP. So,
But this is just for the paralog conservation?
yes.
Is there like the mutational density, the burden score that's correlated with this for non-PER regions?
Well, if you download the table here, well, it will show you.
I'm sorry, maybe it's not loading on here.
Yeah, no. If you download the results here on that button, you will see the burden measurement for all of the sites. Yeah, so you can make your own plot if you want to. We try to be really transparent about that, so you can get all the numbers. Then I don't know. We have time. Yes. The final example is this one, CRAS S17. And this is an example that it's related to a thing that I already told you, but it's good to also stress out. We're on the parallel side, but if we change to the Missense burden, you try the S17, and it belongs to a PER, but no disease association there. So why is that? It's because it's surrounded by other disease associations that you will not see if you evaluate just the variant. That's also something important to consider. Let's say if we go back, I can show you that more clearly. And we have the complete alignment here. I took a screenshot of this. There. This is the table mode of CRAS S17. There it is, S17. No reports on CleanVar or any other patient repositories. But when you consider all the family members, you will see that it will cluster a lot of disease associations from other family members. Do we have Sumaiya? She left? Okay, but it was until 2:15, right?
Actually, I have a question.
Yeah.
How do you treat isoforms?
We choose the canonical one, according to Ensemble. If we, that's something that reviewers raise up because you can have associations on specific transcript, and that's possible. It's a trade-off. But if we include transcript on the family alignment, multiple testing will kill all the results, because you have to align all the combinations of transcripts with each other, and when you test the window, you won't see any association. All of these PRs are adjusted, Bonferroni corrected, so they are past that. But if you start to make combinations of all the alignments, that adjustment will actually kill the results.
I have one more question. Have you ever looked at species conservation for a single gene?
No, we haven't. For that, we will need to have variants, disease-associated variants for all the species to make the burden. I'm not sure if that's available, but no, we haven't tried.
Yeah, I was thinking more for just population variants, because I've looked at it briefly and definitely seen a high correlation with population variants in regions that differ across species, which makes sense.
Yeah. I will expect that too. So the conservation of paralogs can also happen on orthologs. That's and GERB, which is the score that measures conservation across species, is pretty successful in detecting these key regions too. It's kind of old, but still is being used a lot. No. I have a, but do you have a?
Oh, no, I was wondering if you could
No, it's just that the next part, it's a bit moving this into the ACMG guidelines, because I've just showed you how to use PER, but when it comes to making the call of pathogenicity, you need a lot of more arguments in this. So for that, I would like to introduce the online judge interval. Yes?
Is there a way that we can download those scores from the website?
Yes, you can download individual family alignments or the whole results from GitHub. They are all available. You have the families, the scores, the alignments, gene-wise files, so everything's there. It's even available the R code to make the plot, like just a replication of the site. So that's also for people to play around. So now coming back to variant interpretation, the online judge, interval. Does anybody here knows interval? Well, interval it's the automatic implementation of ACMG guidelines. So all of these criteria that were a lot of them and with different combinations to make a call of pathogenicity were coded and put into an online tool that it's available for everybody to use. So it's designed to help and interpret clinical data. It's easy to use and almost automated, because every variant interpretation needs a manual curation. That's the whole idea of this, to make we need the judge in this process. We cannot make it automatic. And it's a command line tool too, so you can annotate with interval a lot of variants if you have them. How it works is just that it grabs all the arguments from interval and then annotates them with all the information available on ANNOVAR, which is so you put their exome sequencing project, you put ClinVar variants there, and all the arguments that can be computationally annotated, you can do that immediately. And then you get these results and you get the opportunity to manually interpret your variant, because you will have more information. You know that the variant is de novo or not. That's something that the judge is only able to know if parent information is available. You won't find parents' information on the database, just as an example. So this is the address of interval. I invite you to visit the tool, because here we will find, on top of all of the things that I've shown you, you will find how to use PER in combination with everything else that we know. We will find the PM1 argument to modify if the variance belongs to a PER. So the front end of the site looks like this. Is anyone seen? Oh, I'm not presenting, sorry. Better, not better, but let's come back to the interval site. Now this is going to take too long. And well, interval. Okay, this is the front end. You have 3 ways of input genetic variants, but this is how you can use interval. You can start to build cases. So case 2, a boy 6 months age, febrile seizures, diagnostic unclear. So this can get pretty clinical. You have a panel sequencing variant that you need to interpret. This is a variant that you can put directly on interval. You know that the mother is positive but unaffected. And I invite you to put this variant into the site. Yes? Were you able to input the variant on interval and make a call? If we can do it together too. So let's see. intervar. This is from the same group that produced Anovar, so it relies on it. You put the chromosome, then the position, and then G by T. So you send, it's in Spanish. You get this, right?
Yep.
Yeah. You get uncertain significance. But you can do, here's where the judge work comes in, because all of this was automatic, but then if you press on details and adjust, you will get the 28 criteria from ACMG guidelines. So, do we know the variant is de novo? Highly likely, yeah, right? Oh no, it's present on the mother. Yeah, it's not de novo. So we cannot click this. Having the 28 arguments of ACMG guidelines here shows you, gives you the ability to modify all of them, but the site recommends to modify the blue ones, where we can have personal information about the patient. So you have to pay attention to all of these arguments. So just to wrap it up, because I think I'm a bit short on time, if the variant was de novo, you can press here, and then you can recalculate the interpretation. So now we have a variant that was initially of uncertain significance, but since we knew that it's de novo, we integrate that into the analysis, and we get that the variant is now likely pathogenic. But we can know more things. If you go back, then how about this if this variant, as I showed you earlier, belongs to a PR? In the hypothetical case that it belongs to a PR, oh, it's already clicked. Okay, that's good. But if it belongs to a PR, you click here and recall your variant of, recall the interpretation, and so on. So this is the final stage of the variant interpretation process. Every score that you know can be used here with different purposes. PRs, as I showed you, can be used as a PE1, ClinVar as a strong argument for pathogenicity, and so on. The combinations of these are quite complicated on the background to call pathogenicity. It's like almost like a poker game. You have like a deck of cards with arguments, and then in combination, one of them wins, and it makes a call. And if you use intervar for this, you can make the clinical interpretation a lot more easier. So, well, all of this has been 2D variant interpretation, and now Sumaiya is going to present you what we can do on 3D. And also, all the things that Sumaiya is going to show you can be integrated again into intervar on the arguments that you will explore. I invite you to read the paper and study all the arguments, how you can acquire more information about this, and a big thank you to all the people involved in this. So, thank you.
Whether or not there's an API for this tool as well? What if I want to get everything?
If you would like to get everything, you can get it from...
In a programmatic context, like batch mode or better.
Well, I would advise you to use the BED format of all the variants. It's available on GitHub. Maybe I can show you the GitHub. Yeah, it's a bit GitHub. So we have LAL group here, and PRs are there. And then you have a tutorial with some examples. You have a gene-wise analysis of SOX9. You can have the analysis of glutamate receptors and voltage-gated sodium channels. The tutorial is designed for you to replicate exactly what you see on the tool with the code and everything. And then on the database, you can have all the family-wise and gene-wise results, the Missense variants that I use as an input from Nomad, the variants that I use from CleanVar, everything. Even the amino acid code that is really necessary for all of this. And yeah, everything's there and it's available for download and yeah, if something is not there, feel free to
that kind of use case, we want to set up our own kind of instance of it, like by downloading these, right? If I want something more immediately transactional.
You can grab the image of the BroadE and launch it. Yeah, that's already there. It's because the site, as you can see, it has a BroadE domain, so the image is there. I think it's available to everybody. And the other important thing is that if you have your own alignment, let's say if you want to align more genes because you're interested in a certain domain, the framework works perfectly. So you can put a clustal file here, which is you can get from muscle. Yep. This is muscle output, and you can integrate that into the pipeline, and you will get PRs or not. That's prepared to do that.
So we are now done with our interpretation of variants on protein sequence. We'll now go to the structure. So just to remind you with the background where we were, I'm sorry, but is there any echo or something happening? No, right?
We can live with that.
Is there any echo?
It makes you sound like you're in a train car.
But this is down.
Yeah, it's soft.
All right. Yeah, so we'll carry on. All right, so we were interpreting missense variants, right? So missense variants propagates from gene to sequence and then to structure. And as Eduardo explained, and I also initially said that a missense variants can be benign to uncertain significance and to pathogenic. And there is a specific community that sets up some guideline, that is American College of Medical Genetics and Genomics. Guidelines that through some criterion, they categorize a variant as a benign variant, likely benign variant, a variant of uncertain significance, likely pathogenic and pathogenic. So saying that, first I will show you that what was the hypothesis behind this work. So given this cartoon example, you can see that in this gene, we have, we assume that there is a single nucleotide variation here, and when we map this variation to protein structure, then we see that this variation is changing the amino acid in this position in the structure. And then let's say we have another mutation in here, in gene, and when we map it to the protein structure, then we see that this mutation is impacting this particular amino acid, which just by visually inspecting, we can see that it's near to this sodium ion and some kind of ligand is there too that I don't know yet, but it is located, let's say, in possibly in a critical region, right? Just by visually seeing, we can tell. So that's why we thought that it is possible to link these two fields, to link underlying cause to the molecular effect. So the hypothesis was variant interpretation can be done using the protein structure, because after mapping, we may be able to say that this specific variant is perturbing the protein stabilizing interacting network, and that is why this variation is basically pathogenic, and the other variation is not pathogenic, but it's a benign variant. So this is why we thought that now it's the time that we integrate these two fields together under this hypothesis, right? And with that, there are some set of rationale also behind this work, and one important one is this PM1 criteria for variant interpretation, that Eduardo also said. So now let's read out this guideline together, that PM1, mutational hotspots and or critical and well-established functional domain. Now, at least I have faced many questions in last 2 years that what is the definition of this domain here? Many questions. And actually, what is even the definition of mutational hotspots? Who define in a particular way what is a mutational hotspot? Okay, let's read a little bit detail. Certain protein domains are known to be critical to protein function, and all missense variants in these domains are identified to date have shown to be pathogenic, and these domains also lack benign variants. That means we have some more detailed knowledge, right? So it has to be a particular domain, critical for function, now who knows? And then in that domain, there should be an enrichment of classified pathogenic variants, and this domain also should lack benign variants, right? That means there should be, we should be able to see a cluster. And that is how we identify that this is the hotspots. Now, we have some other segments of this criteria, that in addition, mutational hotspots in less well-characterized region of genes are reported, in which there were all the variants that were reported were pathogenic. That means it doesn't necessary that it will be a functional domain. It can be a less well-characterized region that is somewhere flanking in the protein structure that we never considered that they are important, but they might be important because all the variants that we see there are basically pathogenic. This description actually tells us something, that this description is not well defined, let's say. Now, we did some preliminary data analysis that we asked a question that, is the protein domain the only critical protein feature to be considered? Now, for domain annotation, at the database that I use is UniProt. That UniProt has this domain annotation, and when UniProt is saying that this is a defined functional domain, I'm calling that, yes, that's a functional domain. So we investigated 1,330 disease-associated genes. Out of those, majority has been implicated in Mendelian disorder, and we found that 30% of the pathogenic variants in CleanVar did not fall in any functional domain that was annotated in UniProt. So we had the answer to our first question that no, protein domain is not only the protein feature that we should be looking for. So, with that, the other observation that I had, the first one I just finished, and the second observation, throughout some independent case studies, I have observed that linearly clustered pathogenic variants can cluster in, linearly scattered pathogenic variants can cluster in protein structure. So here what you can see is the linear protein sequence of the calcineurin complex. So it's the gene PPP3CA. And this magenta highlighted amino acids are the positions of de novo mutations. And when we mapped these positions onto protein structure, so here in the zoomed out figure, zoomed in figure, what you can see is these are the 4 positions that are representing these 4 amino acids that are linearly quite scattered, but they are forming basically a metal interacting pocket in the structure. So the second observation is how protein structure can be valuable, because we don't know this well-characterized region that we are never looking at, and possibly in different conformations, they are coming together to perform a specific function that we never considered in interpreting variant. So with this 2 rationale, we asked this question that, okay, so what could be the characteristics, the other characteristics other than functional domain, right, that represents those mutational hotspots. Now, to answer this question, we set out for this project where our objectives were, we will characterize these positions, these locations of pathogenic and benign missense variants on protein structure. We would like to highlight the mutation-induced impact on structure and hypothesize the variant pathogenicity. For an example, we don't want to give the user only a score, showing that this one probably has a score of 0.9 and this one have a score of 0.2, we want to also give them a reason that I'm saying it at 0.9 because it is perturbing an interaction network. So we want to give them a reason. And also, we want to disseminate our results using a user-friendly web-based platform, and that is how MISCAST was born. So these were the three objectives that we wanted to meet under this project. Now, step one of this project is, of course, mapping missense variants onto protein structure. But what you can see here is this arrow is facing here and this arrow is facing here. It's because there is no possible link that could be established between missense variants and 3D structure. So what we had to do is we had to take the variation information onto sequence and structure information onto sequence, and we had to aggregate that from sequence. So we started with these three different databases. For protein structure, we curated a protein data bank, and we collected all human experimentally solved protein structures. For genetic variation, we went to Nomad to collect the population missense variants, and we investigated two pathogenic variation database. One is human gene mutation database, HGMD, and another is CleanVar database. From CleanVar, we collected only pathogenic and likely pathogenic variants. And for HGMD, we collected only the disease mutations. And in combined, we are calling them as a pathogenic variants or a patient missense variants. And our sequence database is UniProt. So this is how we established the link. This is actually the challenge, the reason why I was saying that there is no possible map, because probably you all know that the genomic positions are stored in VCF file in variant call format, where you have your reference nucleotides and then the alternate nucleotides and the positions in human genome. And for the protein data bank file or PDB file, you have the three-dimensional coordinates X, Y, Z for all the atoms of a specific amino acid. So as you can see, there is no possible link whatsoever between these two different file types. So what we did is we used an intermediate database called SIPS, supported by EMBL EBI. And SIPS has a structure to sequence mapping. So for each amino acids on structure, it tells that what is this positions of this amino acid on protein sequence. And for each structure, this mapping is available in SIPS database. So for each of the human protein structure, we collected this mapping from SIPS. So this is how we established this first link. And then we used Ensemble to annotate the variants on protein sequence. And then we combined it in the sequence space.
What's it relative to?
I'm sorry?
You're mapping onto the 3D structure.
Yes, so.
Like don't you need some coordinate?
Yeah, so of course. So this in 3D structure, we are mapping onto the C alpha atom. And that is the amino acid position on structure. And in sequence, we have amino acid position. And in the, for variant, we have the genomic positions, right? But using web, we can get the corresponding amino acids that a variation is mutating in the sequence space, in the protein sequence space.
From the PDB of the C alpha.
Yes.
So if you want to compare two things, they're both going to be relative to the unit cell of that crystal structure.
Yes.
Use the coordinate directly. But so what happens when I have two different crystal structures of the same thing? How am I getting spatial information about the distance between those residues from two different crystal structures?
So if you have two structures, you have to do it separately, first of all. And you have to know that if you are lucky, then your 10th amino acid will be the 20th amino acid in both the structure, if you are lucky. If you are not lucky, then your 10th amino acid.
I'm worried about that case.
Okay.
The amino acid number is different.
All right.
I'm worried about the spatial positions being comparable between them. So if I have a bound and unbound, ligand bound and ligand unbound.
Yeah, so you have two different conformations.
Protein. Unit cells aren't guaranteed to be the same. Therefore, the coordinate reference bases are not guaranteed to be the same.
Sure.
So how can I compare the positions extracted from one of those crystal structures to the positions extracted from the other.
So you map on both, and then you visually compare.
Well, we're not comparing. But I want to already. I already want to based on the description. Version 2.
Yeah, version 2, exactly. So for now, you can map from sequence to all possible structure, ligand bound, legal unbound, if it is available in PDB.
From sequence, I'm not worried about that either. But they're not aligned yet. But if I have those two structures,
Yeah.
are
So, okay, so now what I'm going to show you in the end is how you can extract the structure with variants mapped, and then you align them in PyMol. You do the structure alignment.
do
by,
it after.
yes, yeah, after.
Do the alignment myself after.
Yes.
With the variants already mapped.
Yes, with the variants already mapped. That's good, right? Yeah, that's already good.
It's a variant.
All right.
I will still try to get you to do it for me later.
Sure. Sure, we understand that. No, we appreciate demands. It's the demands that we want to do in the next step, right? So, all right. So yeah, feel free to interrupt like this. No worries. And with this framework, what we could do is for these 1330 genes, for which we had human protein structures and also population and pathogenic variants that were mappable on structure, we established this link. So for 1330 genes, we mapped, excuse me, for around 400,000 population variants on the structure and comparatively low number of pathogenic variants on the structure. But interesting information is we could map less proportion of population variants, but higher proportion of pathogenic variants. And what we guess is it's because our structure solvers, our scientists have always tried to solve the functionally important domains and regions, and that accumulates like a higher proportion of pathogenic variants, right? So it's a bias that's coming from there.
Just to be clear, the reason when you fail to map is because that amino acid is not present in the structure.
Yes. Yeah. So after having this pipeline ready, what we asked is what are the features that can give us structural and functional insights besides protein domain? So we collected a large varieties of protein features that includes 3-class protein secondary structure information. And then we also collected 8-class secondary structure information because we wanted to see that we only see these pointers, spiral shapes, and flexible things, but these spiral shapes can be 3 different types, and their structure is different. Their number of interactions is different, so they can have different contribution. And then next features that we considered is the surface exposure level to solvent. And what we did is we collected the quantitative value of the solvent exposure level, and then we normalized it, and then we discretized it into 5 groups, as you can see that if it is really in the core, or buried, or medium buried, medium exposed, or really on the surface. And the next feature that we explored is protein-protein interaction types, and this feature is coming from PDB-SUM database. And PDB-SUM database again stores the protein-protein interaction for each of the PDB that is available in Protein Data Bank. And of course, and this is also available for protein-protein complexes because these annotations are collected from the surface interactions. All right, and then the 5th feature that we collected is 6 different types of post-translational modification site. And the database that we explored for this is Phosphosite Plus. And from there, we collected that if an amino acid on the structure is modified by adding an acetyl group, so it's acetylation, methylation, phosphorylation, glycosylation, adding of a sumo molecule, that is sumolation, and a small ubiquitin molecule, that is ubiquitination. So all phosphorylation sites are extremely important to have the mature form of the protein structure. So what we hypothesized that either if it perturbs directly the amino acid that are on the post-translational modification sites, or close, like within the 10 angstrom distance, it might have some impact on the post-translational modification site. So we considered the amino acids that are right on the site or within 10 angstrom spatial distance. All right, so with this, these are our structure-based features. I'm sorry, it's not working. So these are all our structure-based features. And then we also collected the physicochemical properties of amino acid. So we grouped if the amino acid is aliphatic amino acid or aromatic amino acid, or if it is just a special amino acid like proline, which is very small, and cysteine having a disulfide group, sulfur group, it's extremely reactive, and then the glycine. I guess glycine is the smallest residue, not proline. Proline misses C beta in the backbone, so it's still somehow flexible. And proline is also an important amino acid because usually proline is considered as a helix breaker. So if proline is mutated, there can be some impact. So we grouped these 3 amino acids with special characteristics as a special group. All right, and the last feature that we explored is from UniProt. So from UniProt, there are several annotations available in UniProt, and actually the total number is 25. So there can be zinc finger, motif, binding region, DNA binding region, nucleotide binding region. We collected all 25 annotations that are available in UniProt, and then we grouped them together as functional site, binding region, functional domain, functional region, or regions that are performing some molecular signaling like peptides, propeptides, or signal peptides, right? So these are our feature space. With this feature space annotated on the amino acid level, now we will basically characterize the pathogenic and population variations on protein structure. So just to give a summarized overview, so we had 7 different main groups of features with 40 different subtypes that I have just shown you in the previous slides. All right, so how to characterize? We did it simply by a statistical analysis, which is called a two-tailed Fisher's exact test. So for each of the feature, we count how many of that feature is present in the amino acids that are affected by population variation, and how many are present for the amino acids affected by pathogenic variation. And we do a comparative analysis to see that what are those features where we have burden of pathogenic mutation compared to the population variation, right? So this is the output that Fisher's test gives us. And then we did it for all 40 features.
How sensitive is the
Boundary?
Yeah, the boundary between a region. So you can imagine, like, for some of the ones that we saw earlier, there's, you know, a very tiny region in between that drops out of significance and then comes back. But this Fisher test requires you to make a hard threshold, right? So how sensitive is it to where exactly you put that?
So 2 answers of the question. First is this characterization is amino acid space. Amino acid-wise characterization, it's not giving a region, first thing. So it can say a specific amino acid as an amino acids where we observe an enrichment of pathogenic variants. And the cutoff for the significance that what we are observing, how significant it is the p-value of the Fisher test. So we have a cutoff for the p-value that gives us the significance level, right? So the region space, the special region space, we will go later.
So you're going to give a score for each amino acid?
We are going to give you a score for each amino acid. Yes. And we'll also show you that what is the reason we are giving that score, because we'll give you the features. We'll show you that if it is perturbing an interaction, if it is perturbing a post-translational modification, we'll show you everything. Okay, let's talk later. Sure. All right, I sometimes works, sometimes doesn't work. Okay, I don't know. So this is the output of the Fisher test. It's literally not visible. That doesn't matter. So let me explain you, give you the overview of this plot. This is a classical forest plot to see the Fisher test output. How to read this plot is this vertical line that you are seeing. This line represents that there is no association between the variant type and the feature. And when these red dots, the circles are on the right side of this vertical line, this represents a feature that has an enrichment of pathogenic variation. And when we have the dots on the left side, then this represents the features where we have the enrichment of population variation. And if it meets the cutoff, then we have the red or blue color. And if it doesn't have the color, if it doesn't meet the significance level, then we don't have the color for it. So you can see that the labels are kind of turned off, right? So in total, out of the 40 features, we had 18 features where we observed significant burden of pathogenic variation, and 14 features where we have significant burden of benign variants. And now let's look a little bit detail into the output. The pointer is not working again. All right. So which feature had the most significant and enrichment of pathogenic variants? So that feature is the disulfide bond. We observed almost 20-fold enrichment of pathogenic variants on disulfide bond in protein-protein surface between two cysteines. So this is the most significant features, and it makes sense, right? All the structural biologists or everyone who is here, I guess it makes sense that it can be even more significant than domain, for which sometimes we don't know anything. Yes, question? Okay. All right, and then the next two highest enrichment that we observed for those features are two post-translational modification sites. So as I have explained, like the disulfide bond makes sense because we knew that disulfide bond is between two cysteines that are covalent bond and extremely important for the stability of the protein structure. And when that bond is perturbed, we can say that there will be a major destabilization of the structure. And when post-translational modification gets, the amino acids gets modified, then possibly it will not allow this modification to happen, and that may also lead to the altered protein function. So this observation already gives us an idea that there are other features that are more important than protein domain that possibly we are not possibly, definitely we are not including in our variant interpretation now, right? So we can incorporate this, we can incorporate this own judgment that Eduardo was saying in our variant interpretation.
Can I ask a question?
Yeah.
If you go back. So I noticed the features positively charged, negatively charged are grayed out here. Does that mean mutation of a positively charged residue to any other amino acid?
I'm sorry, this one?
Yeah, I would just think
Yeah.
that if you change electrostatic interactions, that would be fairly disruptive. But apparently it's not.
No, it's not. At least in this large-scale study, they are not. But what I can tell you is here these amino acids are grouped, right? So you can see that the 3 positively charged residues are coming together. Now, if you split them and do analysis on each 20 natural amino acids, then just because I did it, I know that arginine has a enrichment of pathogenic variants. But here we are considering them as combined. So in combined form, in a group form, they are not coming as a significant pathogenic features. Yes, that's true.
Just to be clear, that's mutation of any one of these 3 to any one of the other ones.
Mutation of only these 3, not any of the other one. Yes.
So, for example, a histidine fibrillin mutation would fall under this.
Yes. Any kind of mutation of histidine, the histidine to alanine or methanoalanine or anything will fall here.
And this is across all 1,300 genes.
Yes, it's for all genes. Yeah, and yeah, this is something that we haven't done. That specific exchanges. Yeah, we did not do that. Yes.
Do you have any hypotheses as to why those 2 post-translational modifications?
So yeah, so it's actually a good question. So what would be a good way to answer your question is to look back and pull down all those genes for which we have the annotation and which gave us this signal to analyze them and see it. But what we can really see, we can tell is sumolation site is much rare than phosphorylation site, right? And yeah, it is true that phosphorylation site is much more available, but we see both population and pathogenic variants on phosphorylation site, but sumolation site is often, if it is present, then the protein is gone. Yeah, and it would be interesting to dig deep into the data. There are so many things that can be done out of these results, really. Yes?
Can you speculate on mutation, for example, the protection there?
Actually, I will not be able to exactly tell, no, but ubiquitination is the only post-translational modification where we observe that it's enriched with population variation. Yeah. Yes?
For your glycosylation, you have one type of glycosylation. Is that just the one that popped up, or did you test all the other?
No, I tested only for this because there was another type of glycosylation that is available in phosphosite, but we had very less power for them. Yeah, so we had to discard that, yes.
Also, the simulations involve incarnate so the size of the modification that you have compared to phosphorylation is much larger.
Yes.
So would that play a role also?
For ubiquitination?
Simulation.
Is much smaller. Yeah, so I would not say that that plays a role. The observation is true that sumolation site is much rare, smaller set, and most likely whenever we are observing a variation on those sites, it's leading to pathogenicity. It's true that phosphorylation sites, we have much larger sample, and we see a kind of balanced distribution of two different types of variation. I don't think that this will be likely that if the sumolation sample size becomes bigger, then we will lose the signal. I don't think so. Yeah.
What fraction of the proteins that you looked at were, for example, membrane proteins? I'm just thinking some of these features may be important membrane proteins and not so much.
I'm just coming to that point very soon. Yeah. So, all right. Going next, yes. So this will now answer your question. So after having this general characterization, right, with all these features, the next question that we asked is, are these protein regions that are enriched in pathogenic variants and their features can be specific to protein functional class? Like, this is something that has never been done, that in smaller scale, it has been done, that people took the variations, mapped on structure, and then they tried to characterize, but there is no large-scale quantitative analysis that shows that these are the important features for kinases, and these are important for transcription factors, and these are important for transporters or receptors. So to achieve that, what we did is, we grouped our 1330 genes into 24 different protein functional class. And then individually for each of these protein functional class, we repeated the same analysis on all of the 40 protein features. So now, what kind of insights that we could gain out of that result? So here, what you can see is side by side results for all genes and results for kinases. Now, to highlight some specific observations. So for some features, now coming to the sumolation side, again, back, we see that the enrichment of the pathogenic variations has a huge elevation. So if you see that here the sumolation sites were 5-fold enriched with pathogenic variants, here it is 15-fold enriched with pathogenic variants, which basically indicates that when we see kinase, then this feature is even more crucial than the general characteristics. First observation. Second, we can see completely deviating pattern. So considering all genes, disulfide bond was the most significant feature, right? And now when we see in kinase, the disulfide bond had no significant enrichment. Actually, kinase didn't have any annotation of disulfide bond at all. Instead, for kinase, what we observe is now this hydrogen bond and the salt bridge interactions, which were not significant for pathogenic variants in general, now became significant for kinases only. So this is literally kinase-specific characterization of pathogenic mutations. And also we observed some features that are really consistently important for protein fitness for any type of protein, and that is the residue exposure level. So you can see that the trend is always, when it is inside the protein, the trend is going up, and when it is more outside of the protein, it's going down. So what we observed is, by this class-specific characterization, we could summarize or recapitulate those conserved structural regions that are important for function, and also those diversified structural regions that are important for specific function.
Yes.
Yes.
So there was no class where exposed residues went the other way, like in proteins, like scaffold proteins.
Yeah. Yeah.
Like that are known to just interact with other partners.
Yes. Yes.
I would expect their exposed residues to maybe be more impacted.
More enriched for pathogenic variants. Yes, so at least in my characterization, I did not see any protein class has significant enrichment of pathogenic variants. There were 2 protein classes, I don't exactly recall now their name, where we see an enrichment of pathogenic variants, but they did not meet the p-value significance. But I also have to say that our p-value significance cutoff is extremely stringent for this analysis. So if you got this analysis completely, then we had this 24 protein class and 1 general characterization, so 25 analysis for 40 features. So 25 multiplied by 40, and we divided 0.05, which is the normal threshold, by 25 multiplied by 40. So the threshold was 5 e to the power minus 5. So it just didn't meet the p-value significance cutoff.
In general, when you look at this data, she's looking at all exposed residues and all kinases, but what you might be thinking of are just a few key residues that are key for protein-protein interactions.
So like averaging out.
Yeah, so she might be losing it there.
Yeah.
But yeah, there are
kinases, I wouldn't expect that, but like specific classes of proteins that are known to like mediate complex formations or something.
Yeah. All right. So now the question is how we can use these results that we observed for the quantitative interpretation of variance. So we came up with a very simple method. The method could be a little bit complicated, then could be more accurate too, but it's a very simple method. So given a gene and a protein consequence, and this is 417 is the position and the mutation information, what you can collect is, for this 417 position in DDX3X, what are the features, all these features, these 40 features. And then you can ask a question that how many of the features of GLN417 matches with the identified pathogenic variant-enriched feature. And that count is 6. And you can ask this same question that how many of the features of GLN417 matches with the identified population variant-enriched features. And that count is 2. So you subtract them, and then GLN417 has higher number of pathogenic variant-enriched features as informed by structural biology. And through MISCAST web server, what we are saying you is we are giving you this score, and we are also showing you all the features for GLN417, so you can use your own judgment as well. Yes?
Can you elaborate on how we can differentially interpret a neutral score of 1 versus something that's population associated? Like, what does that really mean, population associated?
Sorry, can you repeat the question?
What's the difference between 1, like an odds ratio of 1, and something that's population associated? Is that like pathogenic depletion?
Yes, so this is a two-tailed Fisher test. So 1 means there is no association. This particular feature is associated with none of the variant type. But if the odds ratio is 0.4, that means it's depleted in pathogenic variants and also vice versa, enriched in population variants.
Right, so like how can we interpret that biologically? I guess I'm not thinking of what the implication, it's not intuitive what the implication of that is.
Right, so yeah, so what we could see is biological interpretation would be that feature is likely to be more perturbed by population variance, and you will expect that it might not alter the protein function. That would be the biological interpretation. But if the link between, does that even answer your question?
Sort of. I guess I'm not understanding what the difference, to me then just more left is like lower on this odds ratio.
That means you see more enrichment of population variance on that feature. As left as it goes, that means that our observation of that, like 0.4, it's when you see that threefold, that means it's 3 times more enriched by pathogenic variants and also lack the benign variants. And in the same way, that was the other way around.
So you're still more confident that it's not associated with this?
Yes. Yeah.
Well, yeah, or the variation is tolerated.
Exactly.
Right.
Now there are multiple ways to say that this 3D site, for an example, I like to say that this 3D site is more vulnerable, or this 3D site is more intolerant, or I at least another way to say is this 3D site is a mutational hotspot. Again, coming back to the same.
Could it even be like protected? Like could it be interpreted as like a protected?
Sure, but usually the conservation people use that protected term more. When it is conserved, then it's more protected. Yeah. All right, so now we understood, right, that how we are quantitatively interpreting variant. Now we tested our results in clinically ascertained independent set of pathogenic and benign variants, and we also tested in the output of saturation mutagenesis for 2 different proteins, and we saw that this 3D feature-based characterization is performing quite good. Now the paper is available in bioRxiv, so if anyone interested, this link is also available in MISCAST website. Now, coming back to the showcases of MISCAST. So this is the homepage of MISCAST. It works best in the laptop because it has a lot of structure viewer plugins. And from the homepage, you can see that there are 2 primary buttons. One is variant summary reports and another is variant analysis suite. So we will do hands-on on MISCAST, but I will also walk you through MISCAST here in the slides so that you already have an idea that how it will look like, because it has quite different types of tabs and many other things. So these 2 tabs are for 2 different purposes. The variant summary reports gives you the text and graphical report on the evaluation, so that is the score that I just explained. And variant analysis suite is basically a complete research platform where you can see your variation on sequence and on structure, and you can compare with the identified results, and you can also apply your own judgment. And you can download the structure too with mapped variants. So I'll walk you through. All right, so now coming to the variant summary report page. So this is based on the features of the altered amino acid residue. Here you can first select the gene, and you have a couple of information like which protein it is and from which transcript we are fetching the variation information. And then you have your amino acid residue selection menu. And after you select an amino acid, you can see a score and a report. So I'll go through, right? This is just an overview. And then in the variant analysis suite, you have 2 different types of selection, gene selection and protein class selection. As I have already explained, I have analyzed all 24 different protein classes separately, so you can see class-specific result from this tab. And upon selection of gene, you go inside a page where you have 6 different tabs. The first tab is to show you the general information about the protein, that about the gene, that what protein it is, and then how many of the variation were mappable on the structure and what was the total number of variation available. So what you can see here, for an example, the amino acid residue with pathogenic missense variants is 44, and out of 44, 39 were mappable on structure. And what you can also see is this DDX63X gene is annotated as, of course, all protein class and also as a nucleic acid binding protein. So what is the annotated protein class for this gene? Now in this one-dimensional view, you have the whole protein sequence where the pathogenic variation, population variation, and where you have the overlap mapped on the sequence space. And in the two-dimensional view, okay, sorry, I'm still in one-dimensional view, it's not finished. So I can click, you can click user on a specific amino acid, and you can see all these features that I was just explaining. That here in DDX3X gene, this 417 position that I was explaining from the beginning, it forms a beta sheet in the conformation, and the residue exposure level is 1.5 angstrom, so it's classified as a core amino acid, and it's a polar neutral amino acid, no protein-protein interaction available. There is a post-translational modification sites, phosphorylation sites on the structure, and you can see the structure ID from here. And this specific position is very close, like within 8 angstrom of the post-translational modification site. And also this specific amino acid is part of an annotated region that is interacting, and also a domain. Now, in the two-dimensional view of variant analysis suite, you have this kind of 2D plots, where in the x-axis you see the amino acid residues, and in the y-axis are the accessible surface area values. And also you have the variation positions mapped with red and blue and the orange, the color code that I'm using. Red for pathogenic, blue for population, and orange when we have both types of variation. So often we see this kind of distribution where pathogenic variants tend to be in the packed protein core, and population variants are mostly exposed. Now the three-dimensional visualization tab, you see all the features and the variants mapped in sequence and structure side by side. And these two modules can talk with each other. So if you select something here, you can see the highlighted region on the 3D. So here I have highlighted a domain, which is the ATP, helicase ATP domain. And when I select it, it highlights that domain for me here. And from this drop-down menu to highlight the variant, you can select pathogenic and population, and you can see the positions right on your structure. And from this menu, you can select all possible PTB that are available for this protein. All right. So with that, sorry, I skipped my magic slide. All right, so this is my magic slide that I like to show. So with these 4 different views in MISCAST, that is the summary report, the linear view, 2D view, and 3D view, you can do a comprehensive analysis of missense variants in sequence and structure space, and understand the impact of a variation in the context of protein structure and function, because we are telling you the features and we are showing you the features, right? All right, so with that, I'd like to acknowledge the whole group. Many people were involved and supported me throughout this project. And then, very quickly, I'd like to show that after developing this first platform ready, after having this first platform ready, we have some goals for this year. And what are those goals? And you have already understood that current version of MISCAST has only 1,330 genes, not all the genes because of many restrictions, but let's see what we can possibly do. So we can expand it to the whole human proteome, yes. Now, the whole human proteome contains 20K proteins, and we have only 6.4% covered now in MISCAST, because for only those many genes, we had pathogenic variants and population variants mappable on human protein structures, right? We did not even use structures from other organisms, because we wanted to do the first step very reliable. Now, if we include the homology models, if we do not include the homology models, we still keep the human PDB, but we drop off the restriction of having pathogenic variants mappable, but we can only map the population variants. We could cover up almost 30% of the human proteome. And this is the first step that we will very quickly, I hope, will achieve. Yes, all the features will be mapped in the expanded, the feature space will be the same or even will be more but everything will be more actually. All right. Yes, so that if we include the homolog structure, then we can expand it up to really around 85%. And even if for the rest of the 15% we do not have structure, we can still show you the information on sequence space at least. So next, how we can expand MISCAST is we can really identify the 3D cluster after mapping the pathogenic and population variants on the structure. How? You are seeing here a video where a bubble is really moving around the structure. So what I'm trying to show here is having a target amino acid on the structure, we can define a special bubble and we can do the similar enrichment analysis that we did within the bubble, that if within the bubble we see an enrichment of pathogenic variants or an enrichment of population variants, and we can identify those specific spots on the structure and we can score them and we can map them. And we have done that for individual projects. And one example is here. So this example was for a solute carrier protein, and what you can see is this is the mapped nomad variants, and then the mapped pathogenic variants from CleanVar. Now so some of the spots, so I think I missed some slides, but some of the spots I can show you even from here. So if you see this spot here and this spot here, where we can clearly see that that region is not harboring population variation, but harboring more pathogenic variants. And by doing this special analysis, we can have a quantitative score that are mappable on structure as a heat map. And if this is feasible, then the other interesting thing that is feasible is take the conservation score that Eduardo developed, map it similarly on the structure to see those conserved regions on the structure. And this is actually, this is the PAR score mapped for the protein, mapped for the protein GNI, which is a guanine nucleotide binding protein. And we took the PAR score, and when we mapped it on the structure, we can clearly see this is the part of the region in the structure that are conserved, besides the linear space. And if this can be done, again, what is our really further down the road goal is to map the functional readouts, that is the MIDI-seq data, the HDX data. We have all the experts available in BroadE. So we can have those data and we can map on the structure to, and we can automate this. The magic part of it is it will be all available in the website and you can download and you can do your own analysis. So you don't have to do it manually. Right? All right. So I have explained the goals as well now. With that, thank you very much, and we appreciate your comments. So you can directly send me comments in my email address, and you will find all other contact information, everything in MISCAST. So with that, I finish my slides. I will directly now take you to MISCAST, and before that, I would like to take a 10 minutes break, and therefore I would also like to give you 10 minutes break. Thank you very much for listening to me. All right, I guess let's get started.
All right.
Yeah, we are all energetic, right? Still? Yeah. 30 more minutes. So, can we all access MISCAST?
Yep.
Awesome. Yeah, so patience is our key in these 30 minutes. Nothing loads up, don't move. Wait with a lot of patience. Okay, so as you can see here, I was saying that there are two different tabs. So I will go to the simple tab first. So if we click on the variant summary report, you can select any gene that you want, but I will go for the same one that I was showing you, DDX3X. Okay. So there you go. It's not yelling at me anymore. Now, you have your gene card link here, your UniProt link here. One very important thing to remember, that the only variations that we are pulling for this gene here is from this specific transcript that has to be kept in mind when using the MISCAST. And then amino acid index, I will go for the same amino acid index for 17. And then this is what it shows up. So as you can see that this is a glutamine, the position, and there is no population variance known for this position. And there is one pathogenic variance from CleanVar, and the phenotype is shown as intellectual disability. And as there is no nomad variance here, it's already telling you something about this position, and there is a pathogenic variance. And now what you can see is two small reports, right? So these reports are based on the calculation that I showed you. Let's read the last segment, that the quantitative value that I'm giving you is pathogenic 3D feature index, P3DFI, which is pathogenic variant associated features minus population variant associated features, and then a value. And when this value is positive, then this has more pathogenic tendency. And when it is negative, then it is more likely to be tolerated. This is how we interpret it. And then we have a detailed text report down there. I'm not going to read it now, but what it tells you is something like this, that this amino acid forms a beta sheet, and we found that in nucleic acid binding proteins in our analysis, beta sheet is either a pathogenic feature or a population variant associated feature. So a text description of the comparison with the identified results and the feature of this specific reference amino acid. Is it clear a little bit? Okay, awesome. And now how to read this graph. This graph shows the distribution of the pathogenic variant associated feature and population variant associated features for nucleic acid. This left one is for all protein classes, and then it shows that for this specific position where the count lies. So what you can see is here this count that this matches 6 pathogenic variant associated feature is quite higher than the median, and how it matches with population variant associated feature is quite lower than the median. So again, a graphical interpretation of the score. Now from this page here, from this menu, you can go directly to the variant analysis suite. So I will quickly first go to the protein class tab to show you one example of protein class. So if you type in kinase, just let me know if you are not able to follow. Then in the first information page, what you can see is what are all those annotated genes within this class that we have analyzed. And then what is the total number of pathogenic and population variants that we have analyzed as part of the kinase. And then these features that we found significant for pathogenic variations in kinase, and the features that we found significant for population variations in kinase. Now to see the graphical illustration, you have to go to the next page, which will take some time to load up. So we'll wait with patience. All right, so mine one came. So now you can see for each individual feature, the distribution of the feature in pathogenic and population variants, and also the Fisher's exact test output. The same output that I showed you, but only for 3-class secondary structure in the first row. And then you can see the similar output for 8-class secondary structure, accessible surface area distribution, and physicochemical property distribution, protein-protein interaction types, distance to the post-translational modifications. So this is the combined overview of the protein class, right? not gene-specific analysis. Now with that, we will again go to variant analysis suite. And now we will select a gene. Let's go for CDKL5. You can go for the same gene, DDX3X, it doesn't matter, but I'm just going to CDKL5 now. Now 6 tabs, right? Everyone can see the 6 tabs? All right, so again, in the information page, as I was showing in the slides as well, there are a couple of general information, and then in the all class-based analysis, what are the features that we found significant, and also class-specific analysis output for this specific gene. Now in the one-dimensional visualization, use this bar to see the whole protein sequence. And here the red indicates the amino acids that are affected by pathogenic variants. Blue ones are affected by population variants, and in the orange one, we see both type of variants. So if I select this arginine here, what you can see is we have observed nomad variants here by exome analysis with allele count 3 and allele frequencies here. And we have also seen a pathogenic variants with phenotype encephalopathy in HGMD. Okay, no one other than me talking. Okay, so now here again I'll go down and you can see the 3-class secondary structure features, 8-class secondary structures, physicochemical properties, all the features that we collected you can see for each individual amino acid. And then you can see a comparison. So in this tabular view, what you can see is, in the first column is the feature for this specific amino acid, and in the next 2 columns, the identified pathogenic variant-associated features and population variant-associated features. And when the feature of the affected amino acid matches with the pathogenic variant-associated features, it is highlighted as red. When it matches with the population variant-associated features, it is highlighted as blue. Now, basically from here, you can calculate your score. So this 3 minus 2, the score will be 1 for this reference amino acid. It's pretty straightforward. All right, no question. So I will go to.
So it's interesting. So one region, I think the pathogenic, I think if you go up, it says pi helices under pathogenic, right?
Oh yes, yes.
Pi helix is a very specific type of helix.
Exactly. So pi helix is a very specific type of helices which is more dense. It has more interactions involved. And this was actually one of the striking outcomes that we observed in the analysis, that in general, alpha helices were not enriched with pathogenic variants, but pi helices were. So if we only do 3-class secondary structure analysis, then we will not see any helices coming up as a significant feature. But when we do this 8-class specific analysis, then we see that pi helices, although it's extremely rare, it's enriched with pathogenic variants. And pi helices actually have been studied separately, where it's reported that this residue is conserved and participates in different bindings. So it makes sense as well. So it like kind of goes in line with the literature. All right, so again, this is another page where it will take some time, which is the 2D plots. All right, now again, for 7 main feature groups, 7 different plots, where in the x-axis you have your amino acid residues, and in the y-axis are the different feature types. Good thing about this plot is there is a specific label called no structure. So you can say from this view that from this position 303 leucine to the end, we don't have a structure available. And for the rest, we do have structure, and you can see that you can hover over and you can see the amino acid index and also the specific amino acid, where you have your pathogenic variants, population variants, and both types. Similarly, for 8-class secondary structures, and then accessible surface area, we plot the raw solvent exposure values, and a kind of similar trend is emerging that most of the red dots are down there, and we can see many blue dots on the top. Exceptions are there as well, and also this -25 label here is where there was no structure for the residue.
This kind of gets at James' question, is that like the solvent exposed, there are some pathogenic variants that are solvent exposed at the top, but it's just more population on the surface.
Yeah. Right, then the physicochemical property-based plot, similar, and then protein-protein interaction type, the same. But here we have also the no bond annotation, because while there can be 4 different types of bonds, there can be other protein residues which is still present in the structure, but it just doesn't participate in any bond. So then we have another label called no bond. And then the distance to the post-translational modification site, and again we have only plotted those for which the distance is less than 10 angstrom, as I was saying, because we considered that, yeah, AJ is trying to see very hard, so I zoomed it in. So this specific amino acid has around, let's say, distance 8 angstrom to a acetylation site. And this amino acid phenylalanine at the position 13 is kind of 3 angstrom away from an existing acetylation site. All right. All right. So this plot you can export, you can zoom, and then I'm going to the 3-dimensional visualization tab. So this is the place where we analyze in the sequence and in the structure side by side. So first of all, probably for this gene, we do not have multiple structures available, but if it is available, multiple structures, then you will see everything in this drop-down menu. And then also you will see different structures here. So you can really see that it is often available in PDB, that one structure will cover the first part, another will cover the middle part, another will cover the end part. So you can see which one you want to explore, and you can load it up. And next, the variations can be highlighted in 2 different ways. One way is here, and that is the spherical highlight, that when you highlight, select the pathogenic variants, you can see the sphere positions. And after highlighting, you can also put your mouse here, and you can see that this is the ASN in chain A, and it's in the 140, it's the position in the PDB 4BGQ.
Can you just spin the protein a little?
Yeah, sure.
So it's cool that you can see where that small molecule is. That's where ATP binds.
Yes.
This is a kinase.
Yeah, actually you can expand it, and you can do your full view analysis. If you want to investigate one specific amino acid very close, you select it, and then it comes up as a close view.
So you can see all the pathogenic variants around where ATP binds, which makes sense.
Oh, goodness. Oh, it's coming up very quickly. Not very bad. I think everyone is able to access, right?
Could you highlight the PTM sites?
Yeah. So how you can highlight the features from here is there are two ways. One is if you select this arrow, this little arrow here, then all different types annotations will come in different colors. So it's pretty clear that the coils are coming in green, the purple helices are coming as purple, and the beta sheets are coming as cyan. But if you now want to switch off this coloring, you select again on that specific arrow. Then the selection goes away. And then you can expand this 3-class DSSP-SS, and now you can specifically explore the beta sheets. Now you highlight a specific type, right? And then again, if you select back, click back on this arrow, that selection will be gone. So now, as AJ was showing, we can highlight the phosphorylation sites. One is coming up here, and another is on the other side, right there, here. All right, and then other thing that I like to do, for an example, I like to highlight a specific region, like let's say the nucleotide phosphate binding region, and then in parallel, I want to map the variants on it. Now I can see what are those mutations that are on the nucleotide phosphate binding region. So you can see your variation position in the context of the feature, right? Yeah, all right. So I probably have finished showing everything from here. There are some, there is a snap that you can take from here, and you can bring in all the options to see it in as sticks, as spheres, but I recommend not to do it probably online, but you can, if you want, you can do it as well too. So it's the little tool icon that you have to select to bring out those tools to explore online. Yes. Right, so before showing the structure export, because then I will go to my Mac, what I want to quickly show you is these two other tabs that are available. So in the feature tab, what you can see is amino acid wise, all the features and the mutations that I was showing you, they are annotated and they are available in feature in a tabular view that you can download as a text file, and then you have your tab delimited file downloaded for your offline analysis. And this checkbox, if you select this checkbox, okay, from here first. So here is a little drop-down menu from where you can only see the amino acids, let's say, for which we have pathogenic variants. So now we selected this. Now we brought in our amino acids for which we have pathogenic mutations. And let's say you want to see now the details, like what are the phenotypes associated with these mutations. So you can check in a specific column name, and then the detail column pops up. So now you can see the F to S, this is associated with CDKL5 disorder. And this E to G, it is associated with early infantile epileptic encephalopathy. And this is similar way for population mutation. If you highlight the details, then the allele count, allele frequency, and all those information comes up for each individual mutations. In the index table, what we have given is, again, a little drop-down menu where you can select the amino acids with structure. And here you have your genomic index to protein sequence to structure mapping. So now you can see that this specific position in the human genome, it corresponds to this specific protein amino acids in protein sequence, and that corresponds to this 10th amino acid in the structure. All right, so with that, I'll go back now to the 3D visualization tab, and what I will do is I'll click on this little export button, and then I have a file. You will all have a file. You may have a notification that this can be harmful for your computer. Believe me, it's not. So just keep it. So this is a .py file. So whoever codes in Python, the computer will try to open it in Python, but don't. This is meant to be opened in Palma. So even I will go to the other softwares installed and I will open it up in Palma. So for the folks who are familiar with Palma, you know that there is this vertical bar here where all the selections can be saved. If you use this little bar down there and pull it to the left to see the full selection, then it will be helpful for you. Down there on the right, a little vertical bar. You don't find it? Of course, here. Sure. So after exporting in Palma, first thing I like to do is I like to see it in white. It's just my choice. And then also the other thing that I would like to do, I like to do is always I like to hide all the waters. So all these red dots are gone. And now from these selections that are saved on the right, now you can expand. So in each of the group, in the variance group, as you can see, there are 6 tabs, 2 for pathogenic, and you have separate selections for of full residue and for C alpha only. Because sometimes the chemists and the structural biologists, they like to see it on C alpha. So when it is on the full residue, I don't mind just to color on the cartoon. So these are the positions in the cartoon that have pathogenic variants.
So Sumaiya, for those that aren't familiar with Palma, can you just explain what the groups are?
Sure. Yeah, feel free to ask questions. These groups are the selections of amino acids that has this specific property. And they were exactly the same that you were seeing online.
So she pre-selected everything for you, which is fantastic.
Yeah, and you can bring in your own sequence, and you can see the colors in your sequence as well after you highlight, of course. So the cool thing about having both residue selection and the C alpha selection is, as now I have highlighted the cartoon only, I can just make it green again, and then let's say I want to highlight the C alpha only. I can now show it as spheres, and now I can color my spheres as red. And you can set your scale to make them a little bit smaller. So these are the sphere selection of your pathogenic mutations. So the same thing that I showed you online, how to analyze the variance in the context of the feature. Let me highlight the spheres.
You can really see these pathogenic variants where they're actually in the core of this.
Yeah, exactly. And if you highlight the population ones at the same time, you can see better. So you highlight your population variants and then also color them by blue. Now you can see that the population variants are on the outside and then the red ones are inside.
And the pathogenic is around the ATP binding site as well.
Yeah. So I will now highlight the spheres. Sorry, hide the spheres, because I want to go back to how it was exactly. So now what I like to do is, for an example, I go to a feature, and as you can read it, it's written in detail that this is a nucleotide phosphate binding region, so for full residues. So I like to highlight it in a different color, let's say this cyan color, and now I would like to highlight the pathogenic variants on top of it. another cool thing is, let's say you want to see one specific pathogenic variance, not all together, right? Very reasonable ask. You click on this plus sign, and all the pathogenic variations are individually saved. So now, from these selections, you can highlight only one specific pathogenic variance as a sphere. And that, which one I selected even I forgot, but that one specific pathogenic variance, you can color it in a red, and you can explore. So it's not necessary that you have to always see all pathogenic mutations together. And of course, when you export all these selections and you have this already, you can select your own amino acid that you want to analyze. Let's say you have a mutation in a different position. Now you highlight your own amino acid and see where it falls in the context of all these feature annotations that are available, and also in the context of the classified variants, right?
That's fantastic.
Yeah.
Is for the exposed residues, is there a link for the exposed residues?
Yes. So this is the residue exposure levels, and here you can see the exposed full residues and medium exposed full residues. So I can highlight them in different color. So let me first color this one as white, and then medium exposed full residues as cyan, and then the exposed full residues are magentas. They are like all the outwards here. So one thing to be kept in mind, that this is a very large scale study, and of course already there was a question what to do for multiple structures. So for multiple structures, we had to come up with a consensus annotation. So these annotations that I am showing is based on this specific structure, fine, but we have genes or proteins for which we have 40 structures available, and then we select that if out of 40, 35 is saying that it's a helix, then we annotate is as a helix. And out of all 40 structures, we collect the accessible surface area for each of them, and then we take a median value as a representative accessible surface area. So this has been done to do the statistical analysis, and this is how the features been fetched. So in the expansion that we plan to do on MISCAST, we plan to have the features each and individually for different structures. So it will not be consensus, but it will be representative for that specific structure. So I think that will be even more accurate, right? So this has to be kept in mind. So yeah, with that, any other question? No, it's a very good question, actually. Yeah, so it's not that we do not do personal collaborations, we do. So of course you are welcome. And yeah.
So if there is no structure, and
yes.
you have a homology model,
yeah.
Is it possible to upload in kinematic?
Yes, so these are all within the future plans. I think this is something extremely demanding. And yes, we are trying to develop a personalized module, a version of MISCAST, where we will allow the users to upload their own structure. That will be really cool. Yeah, thank you.
Or score, right?
I'm sorry?
Or score.
Or scores, yeah. Yeah. So these are very good comments. Now the floor is kind of open for your comments and suggestions. So with that, actually, I would like to thank all of you to be present here today and attend this workshop. Thank you very much. Yes.